Hi everyone, welcome. My name is Dr. Moreno and we will be talking about bioenergetics of exercise, part 2. As a reminder, I am a mother of 2, so if you hear them in the background, that is why. Let's dive in. Bioenergetics part 2. We've discussed a lot about ATP, adenosine triphosphates, for 3 phosphate bonds. That's where our energy is stored, right? So where is this ATP molecule stored within our body? 2 places. Primarily, it'll be in the muscle, and second, it'll be in the liver. They have high capacity for glycogen storage, right? They require fast access when the need for it arises. So if we want to stand up and quickly move, we need it readily available close to the muscle. Why do we need it close to the muscle? The muscle is what we utilize. It contracts for us to create movement. So that's why we have it located within the muscle and also into the liver. But the catch is muscle and liver have a very limited amount of storage. Therefore, we don't have excess amount of energy for us to utilize throughout activity. Again, that's another reason why we can't just sprint for hours on end, because we have limited ability and limited amounts of ATP. Glucose and oxygen are needed by the cells to produce large amounts of ATP. What energy system utilizes both glucose and oxygen to produce large amounts of ATP? What do you think? Part 2 of aerobic glycolysis, right? So the aerobic glycolysis as well as our Krebs system. Because muscle contraction and exercise require a constant support and supply of ATP to provide the energy needed for contraction, the metabolic pathway must exist with the capabilities to produce ATP rapidly as well. Muscle cells can produce ATP by any one of the combination of 3 metabolic pathways. For this class, ATP PCR, that is the first energy system to turn on, but we're looking at this energy as it provides us with ATP immediately. Anaerobic glycolysis and aerobic glycolysis, that's where we're starting to produce more ATP together. So how do muscle cells store energy? If we go back to the top of this slide, how do muscle cells store that actual energy? In the form of an ATP molecule itself, as simply put. Bioenergetics includes all various chemical reactions which metabolize carbohydrates. So again, this is the breakdown, a catalyzed reaction of carbohydrates. This can be done for energy for both oxygen and without oxygen. Without oxygen does not mean that we're not breathing. It simply means our muscle does not require oxygen to sustain that specific activity. Those activities are going to be short in duration and high intensities. Your muscle will require oxygen for activities that are longer in duration and a lower intensity. For examples of these, again, go back to part 1 in the first few slides. They'll see that table that gives you a breakdown of different activities and energy contributions between aerobic and anaerobic activity. The second part, synthesize high energy ATP molecules. We know first energy to turn on, ATP PCR. Second energy system to turn on, glycolysis. In order for glycolysis to start, think of a car. What do you need for your car in order to start? You need a key. You need gas. Glucose is our gas. It's part of the whole process to jumpstart this glycolysis process, going from glycolysis, pathway 1, to pathway 2, and eventually to the Krebs cycle, the third energy system. Test question. ETS is not an energy system. It is where we convert all the potential energy. What are the 2 vitamins that we talked about, our shopping carts? NAD and FAD. That's where these get converted in the ETS, the electron transport system. Also, if you read it in textbooks, it could be known as a chain, ETC. Either one, they're the same thing. So ATP PCR. The system provides ATP that is stored in 2 places, readily available for us to use. What are those 2 places? Muscle and liver. Short-term, high-intensity activities. That is what this energy is supplying energy for. Short-term, high-intensity activities. And the start of any exercise, regardless of regardless of intensity. That means we can be getting out of bed, standing up from a chair, getting out of the car, any activity itself. The most rapid process for producing ATP is the ATP PCR system. It involves the donation of our phosphate and its energy bond from the phosphocreatine, aka creatine phosphate, to ADP to form new ATP. Phosphocreatine, right? We have ATP, which is adenosine triphosphate. Now we have PCR, which is phosphocreatine. The phosphocreatine is simply there to help transfer these high energy molecules to produce more ATP. So think of the ATP-ADP cycle. We have 3, which is in blue. We have muscle contraction. We lose a phosphate. We're left with 2. And it continues over and over and over in this process. So again, the phosphates are just the storage of energy in the ATP molecule. We have 3 of them. Creatine phosphates is the transfer of high energy phosphate molecules. Because we have to be able to transfer it from where it's being stored. It can't just be used immediately from that stored location. As quickly as ATP is broken down into ADP plus P, because we have only 2, at the onset of exercise, ATP is resynthesized by that ATP PCR system. Okay? However, muscles store very little amounts of PCR, so the total amount of ATP can be formed is limited by resting amounts of creatine phosphate. 1 molecule of the phosphocreatine leads to resynthesis of 1 molecule of ATP. So in simple terms, a simple 1 enzyme reaction to produce ATP, which is good because the system needs to be fast and quick. We don't want an elongated multiple enzyme step reaction to form ATP. Because we know if we have multiple enzymes, we are only as fast as our weakest link. ATP PCR. The system provides energy for muscular contraction at the initial onset of exercise and during short-term, high-intensity exercise lasting up to 10 seconds. So now we're talking about how long each system actually lasts, meaning how much time, how long does each energy system provide us with energy? ATP PCR provides us with energy from 0 to about 10 seconds. Highly trained individuals can be within 14, but we're going to keep it at 10 to make it a simple, easy, round number. So 0 to 10. Write down ATP PCR system provides energy from 0 to 10 seconds at high intensities. The phosphocreatine reformation requires ATP and only occurs during recovery. At the end of 10 seconds, if you sprint really hard for 10 seconds, will you be exhausted? Absolutely. So after that 10-second sprint, you'll have to recover. This is when we start to reform our ATP and the phosphocreatine to be able to prepare for the next bout of energy. Some examples of the ATP PCR system, predominantly the 50 and the 100 meter sprints, high jump, rapid, intense weightlifting lasting less than 10 seconds, such as a 1 RM, known as a repetition maximum. Any power, forceful movement, football cut, basketball jump for a rebound, those type of movements, that's provided with energy by the ATP PCR system. To your left, in red, the phosphagen system, this is the ATP PCR, provides energy from 0 to 10 seconds. After that, we have the glycolysis system to turn on and then aerobic system to turn on. And we'll talk about how those energy systems supply us with energy as well. What you see here is the overlapping of these three energy systems. We never simply have just one energy system on at one point. All three energy systems are always on. It just depends on the activity as to which energy system is predominantly providing energy at that moment in time. The phosphagen, or ATP PCR system, always turns on first, no matter the activity. But it only supplies us with a very short period of time of energy. Therefore, our glycolysis system has to take over. It provides us with energy for a longer period of time, but not for the long duration. In green, you see our oxidative system, our Krebs cycle. That's the system that you can see that plateaus and can provide energy for hours on end as needed. ATP PCR during sprinting. What is happening? We know during a sprint we get exhausted, but why? We lose energy. As ATP gets broken down, we also are losing that phosphocreatine, which is the high transfer or the ability to transfer those high ATP molecules. So if we don't have phosphocreatine, we can't transfer ATP anymore. So ATP levels appear to not change dramatically, but they change a little over time. But we really lose a whole lot of PCR and the ability to transfer that energy. So the combination of these leads to exhaustion. Therefore, after that time point, we're going to need to recover for the next bout of activity. Within the human body, we have different types of muscle. For the skeletal muscle, we have type 1 versus type 2. What this is showing is our ATP depletion occurs much faster for our type 2 fibers in comparison to our type 1. For the sake of this PowerPoint, type 2 fibers are our anaerobic muscle fiber. Type 1 is our aerobic. So if we apply what we just learned about the ATP PCR system, that would make sense. Our anaerobic system is short bouts, high intensity activities, which means we're going to deplete ATP rapidly. In comparison to type 1, which is aerobic, we can sustain activity for a longer duration. That's all this slide is really saying. So in summary, anaerobic versus aerobic. The formation of ATP via the PCR pathways and anaerobic glycolysis do not require the use of oxygen, yet the aerobic system does require the use of oxygen in order to form ATP. The second energy system, glycolysis. We've been saying that glycolysis has two pathways, and you can see that down at the bottom. So what is glycolysis? Glycolysis is the breakdown of glucose and forms pyruvate with the production of two molecules of ATP. Pyruvate end product of glycolysis can be used in either anaerobic respiration, if no oxygen is available, or in the aerobic pathway, which yields more usable energy for the cell itself. As you can see, we have pyruvate, Pyruvate, as the intermediate step for both the pathways. That top line, glycolysis, pyruvate to lactic acid, this is our anaerobic glycolysis. The bottom, glycolysis to pyruvate to acetyl-CoA, is our aerobic system. Lactic acid is that burning sensation you may feel in your muscle if you're fatiguing and trying to really push forth through an activity. Can we work through this burning sensation? Absolutely. Training can increase, performance can increase by doing so. Acetyl-CoA is the final step in glycolysis, but also required in order to jumpstart our Krebs cycle. So the cytoplasm is the fluid within our bubble, our cell. So remember, we talked about our bubble last in the previous part 1. If we have a bubble, we have our mitochondria, which is the pill-shaped rod, and inside there we have the mitochondria. Outside of the whole rod itself is fluid, which is called our cytoplasm. That's where our glycolysis takes place. So inside the cell, but not so far inside the cell, so it happens a little bit faster than the Krebs cycle itself. The first step in extracting energy from glucose is known as glycolysis. Glycolysis is sweet and dissolve. It is simply the breakdown of glucose. Again, it occurs in the sarcoplasm, which is just the fluid space within our cell. The anaerobic pathway, which transfers energy from glucose to rejoin our phosphate to our ADP. So this can occur without oxygen, allowing some energy to be generated anaerobically, but not a lot. Remember, anaerobic is more the high intensity, so we don't get a whole lot of energy formed while we are performing. Why? Because it's high intensity activity and we need to use the energy for performance, not using energy to reform new ATP molecules. So there is a multiple step process that breaks down glucose. that we have to invest ATP in order to get ATP back. Anaerobic glycolysis can operate most efficiently from 1 to 2 minutes. So ATP PCR system, we said 0 to 10 seconds. We know at 10 seconds, another energy system must take over. Glycolysis, specifically anaerobic glycolysis. It'll take over from 10 seconds to 30 seconds. Your pace will slow down as a result of this lactic acid buildup. As lactic clearance is not able to keep up with the lactic production, this leads to acidification of the muscle tissue and blood, leading to exhaustion and ceasing of exercise. What is this known as? In part, possibly as fatigue, right? Now, this isn't the cause of fatigue, but it leads to fatigue. Okay? So you'll have glycolysis to pyruvate to a lactic acid for our anaerobic glycolysis. Now, the second energy system has that second pathway, right? It must take over at 30 seconds, which we'll talk about in a minute. Some examples of the anaerobic glycolysis: 400-meter race, 100-meter swim, tennis, soccer, volleyball, basketball, football, involving sprint-like quick movements but not at a maximal intensity. And lastly, our third energy system, our aerobic glycolysis. Aerobic production of our ATP occurs again inside that mitochondria, within that double membrane pill-shaped rod. And is the combination of two interconnected processes: the Krebs cycle, which is the energy system itself, and the electron transport chain, or electron transport system. Again, that is not an energy system per se, but it allows us to form new energy. So the Krebs cycle is also called the citric acid or oxidative system for oxygen, because oxygen is required. Primary function of Krebs cycle is to complete the oxidation, hydrogen removal of carbs, fats, and proteins. NAD and FAD, the vitamins, our B vitamins, are employed as hydrogen transporters. So what's hydrogen really doing? It contains the potential energy in food molecules for the synthesis of ATP, which is our ultimate goal for this entire section. This energy is what is used for the ETC, the electron transport chain, to combine ADP and the phosphate to reform ATP. This goes back to that cycle, the basic ATP-ADP cycle. Oxygen is not actually used during the Krebs cycle, but is the final acceptor at the end. Great test question. Oxygen is required, but not until the very final step in order to convert everything into ATP. This Krebs cycle, over to the right, you see at the very top, acetyl-CoA. And backwards, if we work backwards at the 11 o'clock spot, oxalate, also abbreviated OA, has 4 purple circles. So the 4 purple circles merge with acetyl-CoA, which gives us 4 plus 2, giving us 6, leading to citrate, which is a 6-carbon molecule formed from acetyl-CoA. That is the very first step in the Krebs cycle. From citrate, we go through a number of catalyzed reactions. We have our NAD becomes hydrodized into NADH, and that happens again for a second time. GDP, please note that is just another way of saying ATP, ADP, right? So ADP converts to ATP, which is simply energy. So the GDP and GTP, you can go ahead, if you have it marked out and print it out, and replace it with ADP and ATP. It's exactly the same thing. Then we have FAD. It reacts to our FADH. And finally, lastly, we have NAD to NADH. This is just a little bit more in detail. It starts from glycolysis at the top to pyruvate to acetyl-CoA. So if we just look at this part right here, which pathway are we talking about within glycolysis? Is this aerobic or anaerobic? It is aerobic because we end in acetyl-CoA. So it's aerobic because we end in acetyl-CoA, which merges with our OA, which is the 4 carbon. 4 plus 2 gives us our 6 of citrate. And then again, we go through these catalyzed reactions all the way through this process.